So hi guys, welcome back, and we're going to get into translating our genetic code into a protein. So we're at the translation stage. So here we have a very crudely drawn mRNA sequence, and I'll just label all the bits up like we did in the previous bits. So this would be your UTR, our untranslated bit. This would be our poly-A tail. This would be our other UTR. So obviously this would be your 5'. And this would be your 3'. So this would be your 3' UTR and your 5' UTR. And the cap will be here. It's a very badly drawn cap. And we'll call this the 5' cap. Alrighty then, so we've labelled up our mRNA molecule. Now, we're going to do the fun bit, which is translating into amino acid sequence. Now, I don't actually want you to worry too much about the different colours going on here. We'll get into that in a bit. What we're going to crudely do is label up our molecule. Okay, so the very first one is AUG, and it'll help if I kind of zoom in a little bit. Okie dokie. So we have AUG as our first one. Now AUG always starts off the sequence because AUG, don't forget, when it's translated it's translated in codons, which are 3 nucleotides together. Okay? So 3 nucleotides together, so this is codon 1, is always methionine, and this is coded for by ATG, or AUG obviously in the mRNA molecule, because RNA doesn't carry T. Okay, so that always starts. And importantly, I'm going to scroll right to the end and show you another important molecule. And this is another important codon, and this is UAG, or what would be TAG, and this is a stop. Okay, so this tells them to stop. So I'm going to do this by a signal here, which means stop. Okay, so all the rest of it in between is what makes the kind of like the characteristic amino acids, but these were very important. This is your start, and this is your stop. And everything else in between can vary. Okie dokie. So I'm going to go along and translate these. So UUU would be, that would be TTT, which would be phenylalanine, which is an F. And then I've got GGG as guanine, which is a G, which I think is down here. So I'll show you where I'm coming from. So you have GGG, sorry, no, it's not guanine, it's glycine, which is why I've got a G. You've got CUU, which would be CTT. And the way I'd work this is first base here is a C, the second one is a T, and another one is a T. So CUU and CUU, that's going to be leucine, because here's leucine, and then you've got CTT. And then you have the rest here, so you've got GUG, and that is so you've got G, G, U, T. So that will be G, U, T, which is this one, valine. So I'm going to go ahead and do this for all of them. If you want to skip ahead because you've already translated them and transcribed them yourself, because I do admit this takes me a while, then do feel free. So I'm just going to go ahead and label them up with the ones that I've already got labelled. So you've got GUG, you've got those 3, those 3, those 3, those 3, and those 3, and so GUG. Okay, so I'm sure people have already done it faster than me. I have to admit I do take a while to work these out, but there we have it. So here we have our translated mRNA. So we've just roughly translated that, but that isn't how that happens in the cell at all. So it needs to go off and actually be translated by the ribosome, and that's what's going on here. So what we've essentially just done there by using the codons to translate for an amino acid is exactly what the ribosome does. So the ribosome works its way along the mRNA molecule. So you have your mRNA here, it works from the 5' to the 3' as always, And it works its way down, so you can see that the first one here was glycine, the second one here is serine, and so on and so on. I know that methionine's not here, but it's, you know, it's a very kind of, it's just an example. And these guys, your funny T-shaped guys, are your tRNA molecules. Now, their job is to, as you can see here, they code for the complementary sequence and they bring along the amino acid. And that stands for transfer RNA. Okay. Now, what you'll see interestingly here is that this one is CUC, but it's coding for GLU. Now, GLU is glutamic acid, which is GAG. So we see here that we've got GAG, we've got GAG here, but he doesn't carry GAG, he carries CUC, which is obviously the complementary version of GAG. So everything works in complementary base pairing, and it's the same here. We have CAC and valine, but valine isn't CAC. Valine is GUG, which would be GTG here. So again, it works by complementary base pairing. And obviously this is your growing polypeptide chain. So it keeps adding them, and the ribosome keeps working its way along, just this way, sorry, keeps ticking its way along from the 5' to the 3' end, with tRNAs being added now. You may have noticed that the ribosome has it's split into 3 sections. You've got your A section, you've got your P section, and you've got the E section. Now, I remember this in strange ways. So this one is the one where it all starts. It starts in the A section, so I call this one the LO section, as in hello. See, I knew I'm a bit sad. And then you've got the P section here, which is what I call the polypeptide section. So you've got your polypeptide section here. And then you've got your E, which is a very easy one, exit. No weird LO in here. Okay, so you've got LO, polypeptide section, and exit. So that's the way that I remembered the way in which the tRNA molecules enter the ribosome and then leave. So I've written that all down here for you to fill in. The A site, the P site, and the E site. So we have your LO. You can also call this the processing site. That's okay. Whatever makes you remember. And your exit site. Okay, so once the mRNA has been just transcribed, the 2 units separate, and it's time for protein folding. Now I'm just going to do that in a separate section, so I just thought I'd just kind of go into this a little bit more detail. So your ribosome, this is a ribosome, very funny looking ribosome, but it's a ribosome. Okay, and it's got it's split into 2 parts. You've got 2 subunits. This is your small subunit, and this is the one that reads the mRNA. And this one joins the amino acid to make the polypeptide chain. Okay, so the way I remember this is small, brains, because it's thinking about it, it's doing all the reading, and this one's doing all the doing, so this is brawn. See, it's a weird way of remembering it, but hopefully it makes you remember. So obviously the mRNA molecule gets fed in, and it gets fed in that direction. So you've got the 5' to the 3'. Okay, so I just thought I'd do that in a little bit more detail, so hopefully it kind of makes you understand and remember the ribosome process a little bit better. So we can end it there, that's fine. That's covered the main points of the translation, but I've just done a few extra notes for you in case you find it interesting, and of course, because it's biology, rules get broken all the time. So, just a few extra notes. The ribosome is actually made up of both functional RNAs and proteins. So not just proteins, you've got functional RNAs in there. The RNA inside the ribosome is called the rRNA, which stands for ribosomal RNA. The activity that links the polypeptide chain together is catalysed by an rRNA. So this makes the ribosome a ribozyme. Now a ribozyme is an RNA molecule capable of catalysing biochemical reactions similar to a protein enzyme. So a ribozyme is an RNA pretending to be a protein, or it thinks it's a protein. It's very clever. So there you go. An RNA is a great example of a ribozyme if you've never come across that before. Now I've already mentioned about double-stranded RNA, so rules get broken. Double-stranded RNA exists, so rotavirus. The genetic information can flow from RNA to DNA, and this is often seen in retroviruses, so HIV, SARS, Ebola, influenza, and this is called reverse transcription, and it's facilitated by an enzyme called reverse transcriptase. Now it can generate complementary DNA, so we call this cDNA, from RNA, and by using this method they actually hijack our cellular machinery to replicate themselves and establish an infection, which is something we're actually going to cover a little bit in our practical. We're going to look at a retrovirus, or at least how a retrovirus likes to hijack us. So there are probably a few more, but the rules do hold true most of the time, and that's important. There are always ones that break rules, but the biological rules are there for a reason. It holds up most of the time. And then the other cool thing I want to introduce to you is epigenetics. Now, the kind of transcription and translation process sounds very easy. You want a protein, so you transcribe and you translate and you get your protein. However, there is such things as levels of protein. So epigenetics and gene regulation affect the expression of genes, so how much of a protein is produced. So for example, turning genes on or off, epigenetics, you either have a protein or you don't. Or upregulating, which is increasing expression, or downregulating, which is decreasing expression, small amounts of protein or a lot of protein. So epigenetic changes are often heritable, but there is no change in the gene sequence. So the changes in epigenetics which turns genes off, so we can actually inherit those, which, when you think about it, is all very normal, which I'll go into in a second. So changes in gene expression are normal. So think of the circadian rhythm. In the day, light stimulates many complex biochemical pathways that can downregulate sleepy hormones and upregulate the hormones that make us feel awake, in theory. In the night, the dark stimulates other complex biochemical pathways in our brain, and that downregulates our awake hormones and upregulates our sleepy hormones. Now, it's far more complicated than that, but that's obviously just like a quick overview of gene expression. Epigenetics is also normal. Now, think about the fact that all the DNA in your cells is exactly the same. However, your kidney cell looks different and performs a vastly different function than a neuron. And this is because only the genes needed for the kidney cell and its function are being expressed. The rest are turned off. And examples of this include DNA methylation and histomodification. And it's just a great example of the fact that, if you might have heard the term, junk DNA and things like this. Now, junk DNA often refers to introns, but other DNA that's switched off. So we've got genes that kind of turned off that are no longer in use. And that makes sense when you consider that we share 90% of our genome with a chimp, but we don't look like a chimp. Or most of us don't look like chimps anyway. So this is just a great example of genes being switched off, and they're switched off in a sense permanently, as in when you have offspring, those genes are also switched off for the offspring, and then obviously genes get switched off between cells, like the kidney and the neuron example. So I just thought I'd throw that in there as like a great example of all the other things that can affect gene expression and things like that, aside from transcription and translation, because it's never straightforward. Okay, so the next thing we're going to cover is protein folding, but I'll do that in a separate video to give you a break.